Hello everyone, this is Bioevidence AI, an evidence-aware web application for single-cell and single-nucleus marker gene analysis. Marker genes may be statistically enriched in a cluster, but enrichment alone does not prove cell identity, biological function, or causation. Bioevidence AI helps researchers avoid overstating their conclusion. The data input. Users can work with the bundled synthetic demonstration data or upload their own marker CSV. Uploaded data can be validated, filtered, visualized, and exported. In this public demo, GPT access is restricted to the trusted synthetic data for privacy and cost control. Before interpretation, the application checks required columns, missing values, invalid statistical regions, duplicate rows, and duplicate gene cluster combinations. This prevents unreliable data from silently entering the interpretation workflow. Researchers can select clusters and apply thresholds for adjusted p-value, fold change, and expression percentage. Here I selected the root-like cluster and 5 retained markers. The app produces ranked marker plots, a gene-by-gene heat map, and gene-level expression profiles. These calculations are complicated deterministically before GPT is used. GPT is contacted only after an explicit button click. It does not receive the complete data frame. Instead, it receives a compact validated evidence package containing selected marker statistics, thresholds, validation status, and limitations. The output separates statements into directly supported evidence, causations, interpretations, hypotheses, and unsupported claims. For example, the data may directly support that WUX5 is enriched in the root-like cluster. However, that does not prove that WUX5 controls the cluster's biological identity. The claim audit checks whether a researcher's conclusion is stronger than the available evidence. This claim says that WUX5 controls the biological identity of the root-like cluster. Bioevidence AI identifies the casual wording and unsupported and suggests a safer conclusion. WUX5 is statistically enriched or associated with the cluster, but biological control has not been demonstrated. Users can export a human-readable report, structured provenance, filtered marker tables, and interactive figures. Results may remain available even when AI access is locked. I used OpenAI Codex to build, test, debug, and secure the application. GPT 5.6 Terra provides the constrained structured evidence interpretation. The final application passed 68 automated tests. Bioevidence AI helps researchers communicate exactly what their data supports and what may be inferred cautiously and what still requires experimental validation.